living world which the unaided eye or the naked eye cannot see. So ito no. So in this activity, you are able to learn what are the different parts of the compound microscope and their functions. You also learned how to manipulate or kung papaano gamitin, no? Ano ba yung unang ginagamit na objective, and then what are the different coarse adjustments na ginagamit, no? Mga adjustment knobs, 'di ba? We have two. We have the coarse and we have the fine adjustment. Okay, so later on, we will be reviewing again, refresh ko sa inyo what's the difference between the two adjustments. Okay, so you also learned how to compute for the total magnification. Okay, who can still remember what is the formula in getting the total magnification? Sige, how do we compute? Anyone? Yes, Albert.
Yung, for example, in low power objective, ma'am, 100, 10 times,
Sino talama?
wait lang, ma'am.
Sige, sige.
Ano po? For example, ma'am, in scanner, ma'am, 4 times 10 times magnification is equals to 4 times, ma'am, total magnification. And in
Okay.
low power, okay na, ma'am.
Okay, sige. Thank you, Miss Albert. No, in other
Okay.
words, simple words, ang sinasabi ni Miss Albert is in getting the total magnification of the image, you have to multiply the magnification of the ocular lens, which is usually 10, multiply it to the magnification of the lens na gagamitin, of the objective na gagamitin. So what is the magnification of the scanner? What is the magnification of the scanner?
4 times.
4. And then the LPO is
10.
10 times, while the HPO is
40 po.
40 times. Okay? So again, ang scanner 4 times, ang LPO 10 times, ang HPO 40 times. But that is not yet the total magnification. That is just the magnification of each of the scanner. No? Total magnification, i-multiply nyo pa yun siya sa magnification ng ocular lens, which is 10. So if you will be example, I will be giving you an image or a picture of a microorganism, and then sasabihin ko doon na ito is nasa under LPO, and then ang kanyang ocular lens is 10. So I will be asking you to compute for its total magnification. So compute nyo lang, no? What is the LPO objective, LPO magnification again? 10 times the ocular magnification, which is also 10. So the total magnification will be
100.
Oh,
100
okay.
po, ma'am.
Give you 100. So, okay. So, ano lang, no? Clarify lang natin kasi last time, maraming nagkaroon ng ano tayo dito, no? Confusion kung ano ba talaga yung how to get the total magnification. Okay, so you also learned how to differentiate the objectives in relation yun ano to the size of the object. So hindi na natin ano talaga ito dito. Okay, so let's do it one by one. Okay? So these are the different parts and functions of the compound microscope. So first is we have the ocular or the eyepiece. So ito yung part ng microscope kung saan kayo tumitingin to examine no the specimen in your microscope. So it is inserted into the draw tube. So draw tube, ito yung mahaba, ano? Yung sa taas is na ang ocular lens, and then its body is what we call the draw tube. And then sa draw tube, naka-attach yung ating revolving nose piece. No? Yung revolving nose piece, ito yung iniikot natin to change the objectives from scanner to LPO to HPO. Okay, am I going too fast? Or okay lang?
Goods lang po, ma'am.
Okay lang, ma'am.
Sige. Okay, so that is our ocular or eyepiece. So usually 10 times ang ano nito no, ang magnification, but if you remember during your activity, no, nagbigay ako ng table kung saan naglagay ako ng iba't ibang numbers. So that is not ano naman talaga no kasi 10 times talaga ang ocular or ang eyepiece. So ginawa ko lang yun just to see if you understand how to get the total magnification. So next one is we have the draw tube. So this is the barrel that hold the lenses of the eyepiece, and the objective. So ito yung sinabi ko kanina, ito yung mahaba, no? And it holds, so nasa taas niya ang lens or ang ocular lens, and then nasa baba ang objectives that is also attached to the revolving nose piece. Now let's go to the two different adjustments, no? The adjustment knob. First is we have the coarse adjustment knob. It is a large wheel which moves the body tube up or down to bring the specimen into focus. It is used when low power objective is in place. Remember ha, kapag low power, di ba, when you are manipulating a microscope, hindi pwedeng HPO agad ang gagamitin. So ano nga yung mga tendency kapag HPO agad ang ginamit ninyo? Anong pwedeng mangyari sa slide? Yes, Mr. Kasuyon.
Tendency po mababasag yung slide or may hit ng objectives
Yes,
yung top of
correct.
the slide po.
Yes, correct. So may tendency na mabasag yung inyong slides kasi mahaba yung ano, 'di ba? Focus talaga yung HPO, mahaba yung kanyang objective. So ang dapat nga talaga niyo start tayo sa scanner and then LPO and then kunin niyo yung focus using the different adjustments, no? Using the coarse adjustment since nasa LPO pa. And then kapag nasa focus na, that's the time that you will slowly move the objective to the HPO. Okay, so kapag nasa HPO na, again, you will be manipulating again its adjustment for you to be able to get the sharp focus. So yun, in getting the sharp focus, that is now where the fine adjustment gets in. Letter D, fine adjustment. It is a smaller wheel which bring the specimen to its sharpest focus by moving body tube up or down slightly. Okay? So again, ito din, importante din to. You should know na dapat slightly lang ang pag-move nitong mga adjustment knobs. Hindi pwedeng mabilis ang pagtaas ng tube or mabilis ang pagbaba ng tube. Dapat dahan-dahan lang yung pag-move ninyo sa mga adjustments. Okay, listen carefully. I might ask you sa exam kung on how to properly manipulate the microscope. So this is exactly what I am explaining now. Okay. So that is the fine adjustment knob, no, for you to be able to look at your specimen at its sharpest focus. So next is we have the dust shield, no? Kung sino sa inyo ang nakakita na or nakahawak na talaga ng microscope, as you can see, ang nagkakaroon lang ng dust is only the objectives na is only the lenses na nasa objectives, no? Nasa scanner, nasa LPO, and nasa HPO. Yung nasa taas, nasa ocular lens, surface niya lang yung nagkakadust. But yung nasa loob, no? Yung lens sa loob mismo is hindi nagkakaroon ng dust. It is because meron tayong dust shield. So it is a rounded metal attached to the end of the body tube which protects the objective from dust. So para hindi magkaroon ng dust, ng alikabok ang inyong objectives, so this is the function of the dust shield. So next is we have the revolving nose piece. So we all know what the revolving nose piece, no? It is the part of the microscope that holds the 3 or more, no? 3 or 4 objectives. So ito yung dito tayo humahawak. Ito yung nire-revolve ninyo, no? To change the objective from one to the other. Next is we have the objective. So ito na yun kanina, the scanner, the LPO, the HPO. Okay? And then we have the stage. So this is the flat area where the slides to be examined is placed. So ito yung pinaka-visible na part talaga ng microscope, no? So dyan sa stage makikita ninyo ang may hole dyan, and then meron siyang stage clip, and dyan natin nilalagay yung specimen itself. Okay, so after stage, we have the stage aperture. So ito na yung hole na sinasabi ko, the hole or opening at the center of the stage which admits light from the mirror. So kung walang stage aperture, kung walang hole dyan sa center ng stage, so do you think makikita ninyo kung ano yung specimen ninyo? No, no? Kasi hindi magta-transmit ng light from the mirror to the lenses, to your ocular lens. So that is why it is also very important. So next, we have the stage clips. So it holds the slides in place. So para hindi mahulog yung slides ninyo kahit na you will move your microscope, 'di ba? Minsan you invert, hindi naman totally invert it, but binibend natin ng kaunti ang microscope, especially kapag ang microscope ninyo kailangan ay walang ano, you know, 'di ba yung ibang microscope meron silang switch on and off para magkaroon ng ilaw. So yung ibang microscope kasi, especially yung sad to say, no, especially yung nasa CSM, nasa WIMSU, no, hindi ganoon yung lahat ng microscope. Usually yung microscope doon kailangan natin ng light from the sun or light from artificial artificial things like from your phone, flashlight, no, ganon. So para ma-view niyo yung microscope. So you have to bend the microscope. So kung while bending the microscope to find the right, no, light para makita ninyo ang specimen, kung wala siyang stage clips, so mahuhulog ang slides ninyo. That's why stage clips are also important. Okay, so next we have the arm. So as you all know, in handling or in carrying your microscope, you have to hold it in the arm and in the arm and where?
Base.
Base, ma'am.
Base po, ma'am.
Base. Very good. So next is we have the mirror. So you all know that mirror, it reflects light to the stage aperture, to the end light, no, to the specimen of the slides para makita ninyo. Next, we have the pillar. This is the upright bar which supports and connects the base with the other parts of the microscope. So this is for the support, for connection. Okay. Next, we have the diaphragm. It regulates the amount of light passing through the specimen. It may be in a form of rotating disc with holes of various sizes, or it may be an adjustable iris opening. So ito yun para ma-adjust ninyo yung light na papasok or will pass through the specimen. Okay, so there are some specimens na kailangan ng more light para mas makita talaga. Yung ibang specimen naman, pwede okay na kahit kakaunting light lang, makikita nyo na yung specimen. Okay, so this is where this diaphragm gets in. Next is we have the inclination joint. It allows the microscope to incline at a desired angle. So ito na yun kanina sabi ko. Kailangan, there are some times that you need to bend or to incline your microscope. So through this joint, we are able to do that. And last one is we have the base. So this is the horseshoe shape. So yung nasa baba, no, that also functions for the support and is important while carrying the microscope. So those are again the different parts and functions of a compound microscope. I want you to be familiar with it, ha? Okay, so di ba meron akong I asked you last time to identify the different parts, no? Yung may binigay akong picture ng microscope, remember? May binigay akong picture ng microscope and then I asked each group to label the different parts. Okay, so dapat alam
Yes po,
yun.
ma'am.
Okay, so dapat alam rin yun. Kasi baka lang lalabas. Baka lang. Charot. Okay, so next one is we have the microscope manipulations. Ito na yun in-explain ko kanina, no? When manipulating microscope, first of all, no, kapag nasa school proper kayo, kapag mag-face-to-face na tayo, ang unang-una ninyong gagawin before receiving, no, before using the microscope, upon receiving, you need to check it first. So check for damage kasi hindi lang kayo ang gumagamit ng microscope. It is also being used by different sections. So you check it first kung walang damage, and then kung meron, you have to report it directly to your professor. So kung walang damage, no, before using it, you must clean it, no? Wipe muna with tissue paper or with cotton para matanggal yung mga dust sa labas. And then yun na, no? You put your slides, no, sa stage, and then use stage clips to hold your slides, and then start na kayo with viewing. Okay. So, scanner, low power, and then high power. Hindi pwedeng high power directly. Okay, so this activity, no? So, this is the letter E. So, who can, what's this? Remember kung ano yung ginawa dito sa activity with letter E. Short lang. Explain lang kung anong nangyari, anong naging result with this part of the activity wherein you cut small letter E, ganun. What happened to the letter E when it is viewed under LPO and under HPO? Yes, Albert.
Nag-invert po yung letter E, ma'am. Bumaliktad na siya. Pag nasa slide lang siya, ma'am, it's normal. It's a normal letter E. But when na-view na siya in an LPO ocular lens, in LPO, ma'am, nag-invert siya, tapos medyo nag-zoom in, and then same goes with HPO. Inverted pa rin siya, and then mas nag-zoom in siya tapos mas naging klaro yung mga details talaga ng letter E na.
Okay, yun, no? So, when viewed under the microscope, thank you, Miss Albert, when viewed under the microscope, under LPO, baliktad na yung inyong specimen, no? Baliktad na yung letter E, and then it is zoom, but not naman totally zoom na hindi nyo na makita ang buong letter E. Nakikita pa rin ang buong letter E. But when it comes to the HPO, no? Baliktad pa rin siya, inverted pa rin siya, pero hindi na nakikita ang buong letter E. So it's really zoom to the point na parang nakikita nyo na yung details talaga ng ink, no, na ginamit sa pag-print or pagsulat ng letter E. So that is also what happens to your what's this specimens. When you are viewing it under the microscope, it will always appear the other way around. So kung ang pagkakalagay ninyo sa microscope is going to the 12 o'clock direction, it will view, no? Kapag i-view ninyo under the microscope, it will give you 6 o'clock. So kapag nilagay nyo naman 9 o'clock, it will view or it will give you 3 o'clock view. Okay, it's always the reverse position, no? Kapag going to 6 o'clock siya, so ma-view ninyo going to 12. Pag going to 3 o'clock siya, when you put it under the microscope, you will view it going to 9 o'clock. Okay, so yun lang ang tandaan ninyo for that one. Okay. So this one, no, remember the formula, M total is equals to M ocular times or multiplied to M objective. So M represents the magnification. And then field of vision not included and the care of microscopes. Ito na yun. The scanning objective should be in place. Ito din pala, no, pagkatapos nyong gamitin ang microscope, dapat ilagay ninyo in place. So paano ilagay in place? Kapag when you incline the microscope, no, when you bend it even a little, so after using it, you must return it to its proper position. So, i-move nyo din siya. And then, make sure na malinis ang stage, malinis ang lenses, and also, kapag gumamit kayo hanggang high power objective, so if you are done using it, make sure that you will revolve the objectives to the scanner. Dapat pag saulin nyo sa microscope, nasa scanner position ang objective. Next is the microscope must be clean. The stage clips must be pointing forward. The mirror should be in its vertical position and the parts of the microscope with one hand grasping the arm and the microscope, no, and the other one, no, the other hand supporting the base. That is the proper way of carrying. Yes, that's correct, Paul. No, ganon, ang isang kamay, ganon. Okay, sa baba yung isa, and then yung isa naman is sa arm. Okay, so I think that's all for activity number 1 kasi worksheet na ito siya dito, no? So structures and parts. Okay, so once again, for activity number 1, I want you to be familiarized with the different parts and functions of the microscope, how to handle it, and how to manipulate. And of course, don't forget the formula on how to compute for the total magnification of an image viewed under the microscope. Okay? So, I want you to, okay, sasabihin ko na, not just familiarize, I want you to memorize the different parts of the compound microscope. Okay? Kasi magkakaroon tayo ng identification or labeling. Because this is laboratory.
Noted, ma'am.
This is laboratory, more on ganun ang inyong exam, no? So, expect nyo rin, may ibang laboratory pa ba kayo aside from this? May ibang laboratory class pa ba kayo?
Fields, methods, and in psychology lang po, ma'am.
Okay.
And ito po.
Okay, sige. No, basta mga laboratory from CSM, expect nyo talaga na pag lab class kayo, you will really have mga identifications of pictures or labeling of parts, mga ganun, cells, label the parts, ganun talagang lalabas sa exam. Okay, sige, let's continue with the next activity. Okay, any question for activity number 1? I'm
None so
hoping that
far po,
everyone
ma'am.
is jotting down notes ha, especially when I am saying kung ano yung dapat na pag-aralan. Okay, so I'll share now the next activity, which is activity number 2. Yes, Clyde.
Yung question sa activities, ma'am, hindi na po kasali, no?
I will specify kung meron. If ever during the, mapapansin nyo naman, no, during the review ngayon, during the discussion, if I am not including it, it means, so, no need also. Kasi
Okay, ma'am.
usually kasi yung questions, naku, sinasabi ko na lahat. Usually kasi the questions doon sa guide question is also based lang din naman siya sa concept, 'di ba, ng activity. So if I already explained to you the concept, even though I will not we will not be discussing the guide questions, so you can still answer. Okay, did I answer your question, Clyde?
Yes, ma'am. Thank you po.
Okay. Okay, let's proceed with activity number 2. So, this one, no? Easy lang din ang activity number 2 natin. So, this is the chemical basis of life. So, just remember the 4 macromolecules. Ano nga yun? The 4 macromolecules of the living system is we have the carbs, carbohydrates, lipids, proteins, and the nucleic acids. No? So, in this activity, you learned, no, what are the different or you perform qualitative tests. So when we say qualitative test, ito yung paggamit ng indicators for the presence or absence of each type of macromolecule. So how did you learn? No? Ano ba itong mga indicators? Anong mga indicators ang ginamit dito? So gumamit tayo ng color change. Remember? Meron tayong color for positive, meron tayong color for negative. But ang different lang is the lipids or the fats. So, in which hindi yun color. Di ba? Translucent spot test yun. So, kung may translucent, that is positive. Without translucent, that is negative for fats. Okay, so that is what we mean by indicators or qualitative tests. Okay, so let's start with the first macromolecule. By the way, no, when we say macromolecule, these are large polymer made of repeating monomer units. So macromolecules are classified again into these 4. So the first one is we have the carbohydrates. So ano bang carbohydrates? It is the most abundant in the organic it's the most abundant of the organic compounds. So it is also known as sugar or starches basically. Because carbohydrates are product of photosynthesis, the process by which plants and other organisms use sunlight to transform carbon dioxide and water into food. This compound is essential to all living things, specifically plants, in providing not only the energy that living things need, but also as carbon skeletons for organic compounds and storage components. In short, carbohydrates is very important for living system, for living things, no? So, sa carbohydrates natin kinukuha ang energy na ginagamit natin for our everyday life, no? So, basically, carbohydrates is energy. So carbohydrates can be classified into 4 main groups. So we have here the monosaccharides, and then disaccharides, oligosaccharides, and the polysaccharides. And below here is we have the definition or description of each class or type of carbohydrate. So when we say monosaccharide, ito yung simplest of all sugars. So it contains 3 to 7 carbon atoms in their molecule, in each of their molecule. So the function in plants is energy source for all living things. So bakit sinabing for all living things? Because we eat plants, no? We eat gulay. Kumakain tayo ng gulay, and gulay are plants. So, ang gulay is rich siya in monosaccharides, no? Meron siyang monosaccharides. So, that's why we are also we also need this type of carbohydrate because kailangan natin ng gulay. So examples in plants is we have the glucose and the fructose. So just remember, when we say mono, ibig sabihin isa lang yan siya. But even though it means one, no? Kasi one saccharide, it means meron na siyang 3 to 7 carbon atoms. So don't get confused with this one. So next is we have the disaccharide. So when we say di, it means two. It means dalawang monosaccharide joined together. No, two monosaccharides joined together, it will give us disaccharides. So it is also called double sugar or bivose formed when two monosaccharides are joined together by glycosidic linkage. Don't forget, how do disaccharides join or form? It is by glycosidic linkage. Okay. Oh, I bet lumabas ito sa quiz niyo last time, di ba? Ang answer na glycosidic linkage. Lumabas ba?
Yes, ma'am.
Okay.
Yes, ma'am.
So after the disaccharides is we have the oligosaccharide. So this is a saccharide polymer containing a small number. So typically from 3 to 10 monosaccharides. So 3 to 10 monosaccharides, that is already oligo. Kapag dalawang monosaccharide joined together, that is disaccharides. So it is also referred to oligosaccharides. Okay, oligosaccharide is also known as oligosaccharides. And the last type or class of monosaccharide carbohydrates is we have the polysaccharides. It is a long chains of monosaccharides linked by glycosidic bonds. Okay, so understood? Any question for the type of or class of carbohydrates? Easy lang siya. Okay, so
Gets na, ma'am.
just remember lang yung number of monosaccharides, no? For monosaccharides, 3 to 7 carbon atoms. Disaccharides, it is 2 monosaccharides joined together. Oligosaccharides, it is 3 to 10 monosaccharides joined together. And polysaccharides, that is long chains na. So, when we say long chains, it means more than 10 na siya. Kasi kapag 10 lang siya, 10 monosaccharides joined together, that is oligo. But if it's already 11 and above, monosaccharides joined together, that is the poly. Okay, so next is, we'll go with the different indicators, no? So test for starch. So what we call it, starch iodine test or Lugol's iodine test. So ito din, no? In this part, I want you to memorize and to understand, no, the different macromolecules, the different tests, the positive and the negative. The example, for the starch, no? So starch, test for starch is Lugol's iodine test, and then positive is color. Anong positive niya?
Blue-black po, ma'am.
Blue-black, while if negative, it remains to, okay, this one. Okay, so remain lang, the same lang ang color. So ganun, no? Kailangan memorize nyo yun. So a positive test reveals a blue-black color solution. It's a reaction due to formation of polyiodide chains from the reaction of starch and iodine. A starch in iodine and potassium iodide in water has a light orange-brown color. So ito na yun, ang negative orange-brown, and then ang positive is blue-black solution. So don't forget test for starch is iodine starch, iodine test, or Lugol's iodine test. Next, if you want to test for simple sugars, so you will be using the test called Benedict's test. So what is the positive for this one?
Bluish po, ma'am, solution.
Okay, so it will change, no? Changes from green to orange, red brick, or brick red. So that is the positive. So kung negative, it will remain bluish. Ganun hindi the other way around. Kapag negative, it is bluish, remain lang siya. Kapag positive, from bluish, magiging green, no? Or to orange red or brick red siya. Basta something red. Ganun, orange or red siya, that is positive for sugar. But negative, it will remain bluish. The test No, we call the test for sugar is Benedict's test. Next is we have the protein. So in testing the protein, you will be using the Burey test. So it is also known as Piotrowski's test. So what is the positive? Positive is purple and negative is still blue. Okay? Positive purple, negative blue, that is for the testing the presence of protein or the Burey test. And next one is the lipid. So ito na yun siya, hindi naka-base sa color change, but naka-base sa presence or absence of the translucent spot test. So that is, if you want to test for the presence of fats, you will be using translucent spot test. So kung may ganito, may translucent spot, that is positive for fats. But kung wala, just like the water, so it will not give you the translucent spot. So that is negative for the presence of fats and nucleic acids. So ito na yun. Okay. So where do we get most of the blocks in building blocks of our macromolecules? Carbohydrates, ganun, no? Okay, so food. So although carbohydrates are rich in source of energy, can man survive a diet only with carbohydrates? Yes, but only for a very short time. Hindi pang matagalan, no? So mabubuhay siya, hindi siya agad-agad na mamamatay kung puro carbs ang kakainin niya. But after lang how many months, so magkakaroon na ng complications, no? And it might lead to death. Okay, so what causes the translucent spot in the samples of test for positive? Ito na yung presence of fats, no? So kaya may translucent spot, it's because may mga fats, may lipids. Okay, okay, so hindi na yun. Okay, any question for activity number 2? Okay, so dapat familiarize nyo lang what are the different carbohydrates and then what are the different tests, no, na ginagamit in testing the presence of the different macromolecules. What are the macromolecules again? What are the 4 macromolecules? Yes, Manantil. I can't hear you lang ga. Okay, audible na. Yes. Okay, so those are the 4 macromolecules in the living system. Okay, so any question for activity number 2 before we proceed with 3? Or everything clear?
Clear na po, ma'am.
Clear
Okay,
na po,
so
ma'am.
okay, very good. Let's go to activity number 3. So activity number 3 is all about the cell. So what is cell ba? Ano ba ang cell? It is the basic unit, structure, and function of life. No? So lahat tayo nagsisimula sa cell. So all living things starts with cell. Human being, plants, animals, no, and even the microorganisms. Lahat nagsisimula sa cell. So each type of cell is especially adapted to perform a particular function. All the many cells of the body often differ from each other. All of items have certain basic characteristics that are alike. That's why we have different types of cells, no? So merong different shapes and different functions. The study of cell is called cytology. Remember, if the study of tissue is called histology, the study of cell is called cytology. Okay. Okay, another this one. So among unicellular organisms like the bacteria, the whole body is a single cell, while a multicellular organism's body consists of different cells. No, yun, kapag multicellular, from the word multi, which means more than one, madami, no, just like a human being. But meron din tayong mga organism na unicellular, iisang cell, isang buong cell lang siya. no? Just like the bacteria, but not all bacteria. Meron din mga ano na multi. So the unit of measurement of microscopic structures such as the cells is what we call the micron, which is 0.001 no of the mm or 1 over 100 C of millimeter. But I did not include naman micrometer last time during your quiz. So not na din to. I will not ask you to what's this to convert it anymore. Okay, so in this activity, you were able to identify the different parts and functions of animal cell, classify the animal cells based on shape, organization, and functions, and identify the various types of cell, indicate the parts of the body where they may be found or where they are located. So, ito yung isa sa mga mahabang activity natin, no? So, first one, if we have the cell structure and function. So, in this activity, I want you to memorize the basic parts of the animal cell. Okay? Basic parts of the animal cell. So, may binigay akong picture dito, right? Okay? So, this one. I want you to memorize kung ano yung mga parts dito. Okay? Can you hear me?
Ma'am,
Yes,
pwede
ma'am.
i-ano yan siya, ma'am, i-elaborate further kasi parang ano man, ma'am, parang ano, hindi namin masyado. I mean, nag ano man, nabigyan man namin ng name pero parang iba-iba man yun sa amin.
Okay. Sige. Okay. You mean the ano talaga? Okay, sige. Since hindi ko naman ito nilagay
Opo, ma'am, kasi
sa long
accurate
quiz
po.
ninyo, sige, since hindi ko ito nilagay sa long quiz ninyo, I will be sending na lang the correct talaga answer for this one. Okay? Send ko
Thank
sa
you, ma'am.
GC or
Love
sa
you po.
G-class natin.
Thank you, ma'am.
But it's not part of the it's
Thank
not
you,
part of
ma'am.
the long quiz. So pwede siya. Sige, pag-aralan ninyo para sa midterm exam. Okay, so wait lang, palubat na ako. Ang galing charger.
Ni.